Welcome back, guys. Today we are going to start the discussion on this important topic, urea cycle and disorders. Previously we have discussed the basics of amination metabolism, transaminases and deamination and oxidative deamination. Now we are going to start with this urea cycle and its associated disorders. So what is this urea cycle? Urea cycle, okay, it's also called by three names, also called Krebs-Henselt cycle, Krebs-Henselt cycle, okay, also called ornithine cycle. Ornithine is actually recycled back in this step, so it's called ornithine cycle, and also called urea bicycle. Urea bicycle, okay. Why this is called urea bicycle? See in brief, okay. Now suppose this fragment is our urea cycle, and this fragment is our TCA cycle, okay. Now from urea cycle to TCA cycle goes fumarate. From TCA cycle to urea cycle comes aspartate. That is why this is called our urea bicycle, also called ornithine cycle and Krebs-Henselt cycle. Now moving on, we are going to see the sources of atoms of urea. Sources of atoms of urea. Okay, we know urea is NH2, CO, NH2. Okay. Now the first nitrogen here comes from aspartate. Okay, when the nitrogen comes from aspartate. The carbon of the CO is from carbon dioxide, and this last nitrogen or one of the nitrogen comes from our ammonia. Okay, so the sources are ammonia, carbon dioxide and aspartate. Okay, so eventually we can say compounds consumed in urea cycle are ammonia, aspartate and carbon dioxide. These are compounds from which the atoms of urea are developed. They are consumed in urea cycle. Now urea cycle takes place in liver. Okay, takes place in liver, and organelle is a cytoplasm and mitochondria. Cytoplasm and mitochondria. Okay, yes. Now before moving on to the steps, let's going to see a mnemonic here. These are HUG pathway, hug pathway. Okay, now what is the specialty of this HUG pathway here? H is for heme synthesis. Okay, H is for heme synthesis. Urea is for urea cycle, and G is our gluconeogenesis. Gluconeogenesis. Okay, and P is our here for pyrimidine synthesis. All these heme synthesis, urea cycle, gluconeogenesis and pyrimidine synthesis are interconnected. So this totally is called hug pathway. Okay, the rate limiting step of urea cycle, remember, first of all is this CPS1, carbamyl phosphate synthetase 1. Phosphate synthetase 1. This is the rate limiting step or the enzyme involved in the urea cycle. Finally, we will start the reactions. Okay. Reactions. Okay. Before starting, we have seen here the organelles involved are cytoplasm and mitochondria in the liver. Okay. So we first draw a barrier. Okay. This is our barrier here. Barrier. Inside we have the mitochondria. Here is our mitochondria. Some steps will be taking place. This is our cytoplasm. Some steps will be taking place here. Okay, very simple. Yes. In the mitochondria, we will start. So the two compounds from which the atoms of urea are derived, we know carbon dioxide and ammonia. Carbon dioxide and ammonia combine, okay, to form carbamyl phosphate. And we discussed the rate limiting enzyme is carbamyl phosphate synthetase. So the rate limiting enzyme here or the enzyme here is CPS1, carbamyl phosphate synthetase 1. Rate limiting or pacemaker enzyme. Okay, here 2 ATP is used from ADP. In turn, 2 phosphates are used. Nearly 2 phosphates are used. Synthetase enzyme ATP will be used from ADP. Okay. Yes. Now this carbamyl phosphate is formed. This will come and combine with our ornithine to form the second compound here, citrulline. Citrulline is formed. Okay. What is the enzyme here? Ornithine transcarbamylase. Ornithine transcarbamylase. OTC1 or simple OTC, ornithine transcarbamylase. Okay, ornithine combines with carbamyl phosphate to form citrulline. Okay, yes. Now where this ornithine comes from? Okay, this ornithine actually comes from cytoplasm. Ornithine is here. It is transported from cytoplasm to mitochondria through a transporter here. Okay, this is our ornithine transporter. Ornithine transporter or ornithine permease. Ornithine transporter or ornithine permease. From here it goes and combines to form citrulline, ornithine transcarbamylase. Further, this citrulline formed here gets out from mitochondria to the cytoplasm. Okay. Yes. Now we have seen carbon dioxide and ammonia getting involved. There was a third compound here, aspartate, from which one of the nitrogen was derived. One of the nitrogen of ammonia, urea was derived. Okay. Yes. So how this aspartate comes out? So aspartate comes out from mitochondria to cytoplasm. Here is the transporter here. What is the transporter? Citrin. 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 Aspartate glutamate transporter. Aspartate glutamate transporter. Citrin. Through citrin, aspartate comes out to cytoplasm. Okay. Now this citrulline, this does not require any transporter or any specific transporter, as we know. Okay, it seeps out from mitochondria to the cytoplasm. Now these two combine, citrulline and the aspartate, to form arginosuccinate. argininosuccinate, and the enzyme here is argininosuccinate synthetase. Argininosuccinate synthetase. Okay, as it's a synthetase enzyme, 1 ATP used. 1 ATP is used. 2 AMP. 2 AMP. 2 phosphates are used here. Nearly 2 phosphates are used here to convert 1 ATP to AMP. What is formed? Argininosuccinate. Argininosuccinate synthetase is the enzyme from citrulline and aspartate. Remember these two transporters, very important. Okay, argininosuccinate is formed. Now from here in the cytoplasm, argininosuccinate, this is converted to arginine. What is the enzyme here? Lyase enzyme. Argininosuccinate lyase. Very simple. Here fumarate is given out. Here in this step fumarate is given. To where we have discussed in the bicycle to TCA cycle. Fumarate is given. In this step arginine is formed, and further in the next step, so arginine converted to ornithine by the enzyme involved here is arginase enzyme, arginine to ornithine. Here in this step water molecule is added and urea is released. Here only urea is released and water is added in the presence of arginase enzyme, and subsequently ornithine is formed. This ornithine is replaced back to mitochondria through ornithine transporter or ornithine permease. Ornithine transporter, that is why it was also called ornithine cycle. Okay, ornithine is replaced or recycled. Arginase enzyme, before that argininosuccinate lyase enzyme. Here in this step fumarate is given to TCA cycle. Here in this step urea is released. Two very important step here, and this all taking place in where? Argininosuccinate cytoplasm. The ornithine formed is retaken back, and all this reaction takes place in mitochondria. So there is a division of labor here. Okay, this was the gist of the total urea cycle, and urea is released here in the penultimate step where ornithine is formed from arginine in presence of arginase. Water is added here. Moving on, we will see the energetics. There is a reaction in the body, so energetics will also be involved here. Energetics. Okay, first is our carbamyl phosphate synthetase 1. We have seen 2 ATPs are used. Two ATP, nearly 2 phosphate equivalents. These are phosphate equivalents are used. Now coming to argininosuccinate synthetase enzyme, 1 ATP is used to 1 AMP. Here also nearly 2 phosphate equivalents are used. Okay, so from one urea cycle, in one urea cycle to be precise, it actually uses energy in one urea cycle. 4 high energy phosphates are used. Phosphates are used. So in one urea cycle 4 high energy phosphates are used. Okay, so common question asked is how many ATPs are used. Okay, so how many ATPs are used? 4 ATPs, or sorry, 3 ATPs are directly used. 2 here, 1 here, 3 ATPs are used. Okay, 3 ATPs directly. Another question is asked how many phosphate equivalents are used, or ATP equivalents are used. But we'll say 4 ATP equivalents. So we need to see the word ATP or ATP equivalent. ATPs are 3 ATPs, equivalents are 4 ATP equivalents. Okay, now I'm going to see the regulation here. The regulation. Okay, first is our dietary regulation. If we increased our protein intake, induces urea cycle enzymes. Induces urea cycle enzymes. Okay, this is a dietary. Allosteric regulation. Allosteric. Okay, there is N-acetyl glutamate. N-acetyl glutamate. Okay, is a positive allosteric regulator. A positive allosteric regulator of our rate limiting enzyme CPS1, carbamyl phosphatase synthetase 1. Okay, N-acetyl glutamate, NAG, is a positive allosteric regulator of CPS1. Okay, yes. Moving on to we have a compartmentation. The regulation compartmentation. Okay, the compartmentation partly in mitochondria and partly in cytoplasm. This was the case we have seen. Here the inhibitory effect of fumarate. The presence of arginosuccinate lyase enzyme, inhibitory effect of fumarate on its own formation. This is a very important point. Arginosuccinate lies in cytoplasm. So the reason why arginosuccinate lies in cytoplasm. Okay, whereas fumarate in mitochondria. Fumarate produced post to TC cycle takes place in mitochondria. So fumarate lies in mitochondria, whereas arginosuccinate lies in cytoplasm. Okay, this was our urea cycle, its energetics and its regulation. Now we will move on to urea cycle and its disorders. Disorders of urea cycle. Yes, so starting on, first is we have the CPS1 deficiency. With the CPS1 forming the carbonyl phosphate is deficient. This will lead to hyperammonemia. Hyperammonemia. What is the type? Type 1. Okay, hyperammonemia type 1, deficiency of CPS1. Next enzyme in the process, OTC, ornithine transcarbamylase deficiency. This will lead to our hyperammonemia. This is our type 2. Okay, type 2. Yes. Next is the third enzyme in the step, is a citrulline transporter defect. Remember the citrulline transporter or permease, citrulline transporter or permease defect. Yes, this is the one we'll discuss. This deficiency lead to citrullinemia. Citrullinemia type 2. Citrullinemia type 2. Remember type 2, not type 1, type 2. This is also called our citrin. Yes. Through which citrulline and aspartate were transported. Okay. Citrullinemia type 2. The fourth enzyme in the reaction. Moving on, we have the argininosuccinate synthetase. Argininosuccinate synthetase defect, deficiency. This will lead to citrullinemia type 1. Citrullinemia type 1. Citrulline deficiency type 2. Important, remember here. Argininosuccinate synthetase deficiency type 1. Yes. Moving on in the next enzyme. Next enzyme defect, argininosuccinate lyase. Argininosuccinate lyase enzyme defect. Now the lyase enzyme defect will lead to accumulation of argininosuccinate. So it will lead to argininosuccinic aciduria. Argininosuccinate is accumulated. Argininosuccinate is accumulated. Very simple. This is deficiency. Argininosuccinate will be accumulated. Arginosuccinic aciduria. Sixth enzyme on the go is our arginase enzyme. Lyase enzyme, after which is our arginase enzyme. This deficiency will lead to which? Argininemia. Argininemia. Let me write this again. Argininemia. Argininosuccinic aciduria. This was argininemia. Arginase defect. And finally, we have our ornithine transporter defect. Ornithine, which is formed, will be transported back to mitochondria. Transporter defect. We have two transporter defects. That's why I have told you to visualize them properly. This will lead to our HHH syndrome. What is this HHH syndrome? We'll discuss further when we'll see in details. Okay, remember ornithine transporter defect, HHH syndrome. Yes, so we'll see all the enzymes and transporter defects here and what all diseases it will lead to. So let's discuss them one by one. First is a hyperammonemia. Hyperammonemia. Okay, and its relation to plasma glucose. So hyperammonemia is increased ammonia. And in the first step, CPS1 is defect. So ammonia, which is reacting with carbon dioxide, is accumulated, which will lead glutamate getting converted to glutamine. Seen in the general amino acid metabolism video, glutamate will convert to glutamine and the glutamine increases. Okay, now we have two types of hyperammonemia. Hyperammonemia type 1 due to CPS1 defect, hyperammonemia type 2 defect due to OTC defect, ornithine transcarbamylase defect. Here in the comparison cases, NH3 is always more than NH3. Here ammonia type 1 has more ammonia. Also plasma glutamine level is also high here. Glutamine is also higher than this glutamine level here. Okay, so this is not glucose, this is glutamine. Glutamine. Okay, yes. Type 1, all the factors will be more than type 2. Yes. So moving on to type 2 hyperammonemia. So there is question, why increased uracil excretion in hyperammonemia type 2? There is an increased uracil excretion. So the enzyme defect here is ornithine transcarbamylase 1. Okay, so this was responsible for formation of citrulline from ornithine and carbamyl phosphatase. So carbamyl phosphate is accumulated, okay, and seeps into cytoplasm This is a carbamyl phosphate along with ornithine. It was coming from the cytoplasm to form citrulline. Enzyme here, carbamyl phosphate is ornithine transcarbamylase is defect here. So this will get accumulated and seeps to the cytoplasm. And orotic aciduria occurs. Yes. Now, as we are discussing hyperammonemia type 2, we'll discuss the characteristics here. Some characteristics. Yes. First is, it is a X-linked partially dominant. Okay, so only males are affected. Second point is our increased orotic acid secretion. As we have seen, orotic acid occurs, increased orotic acid secretion. Yes. Third one, type 2 is the most common urea cycle disorder. Type 2 hyperammonemia is the most common urea cycle disorder. Type 2, not type 1. Usually we get confused. Type 2 hyperammonemia is the most common urea cycle disorder. Orotic aciduria occurs and it's X-linked, partially dominant, only males are affected. Yes. Now we will move on to arachinosuccinic aciduria. Arachinosuccinic aciduria. We'll see some features, some small features of every disorder, and we'll move on. Okay. And the enzyme here is lyase enzyme deficiency. Arachinosuccinic lyase deficiency. Okay. Feature here is Trichorhexis nodessa. What is the feature here? Friable tufted hair. hair. Trichorhexis nodisa, friable tufted hair in hydrogenous aciduria, lyase deficiency enzyme. Now we'll see the triple H syndrome that we are discussing. Triple H syndrome due to ornithine transporter defect from cytoplasm to mitochondria. Yes. So this triple S stands for hyperammonemia, second S stands for hyperornithemia, or urea. Okay. Third S stands for homocitrullinemia, triple H syndrome. Hyperammonemia, ammonia obviously accumulates. Hyperornithinemia, ornithine as it can't get transported, so it accumulates. Also citrulline accumulates, homocitrulline accumulates, homocitrullinemia. Okay. Yes. How this homocitrulline found? Homocitrulline, it gets found by this carbamyl phosphatase. Yes, that was getting accumulated. It reacts with lysine. Okay, to form homocitrulline. Yes, carbamyl phosphate combines with member lysine to form homocitrulline. Triple H, hyperammonemia, hyperornithinemia, homocitrullinemia syndrome, ornithine transporter defect. Yes. In arginenemia, we know due to arginase defect. Yes. So we'll see the common clinical features of urea cycle disorders. Yes, common clinical features of urea cycle disorders. First is we have obviously hyperammonemia. Yes. Second, we have due to increased ammonia. Okay, there is hyperventilation. This will lead to a tachypnea. Increased respiration lead to CO2 washout. Tachypnea, hyperventilation will obviously lead to CO2 washout. Yes, CO2 washout. This CO2 washout will ultimately lead to a respiratory alkalosis. Yes. And increased ammonia, hyperventilation, respiratory alkalosis due to CO2 washout. Yes. Third feature is our encephalopathy. Yes, encephalopathy will also occur. Yes. And usually neonatal age group, okay, they present with feeding difficulty, lethargy, okay, vomiting from constipation. Most important feature here will be tachypnea. Yes, this is the most important feature out here. It's tachypnea. Tachypnea will be the most important feature here in the neonatal age group, which will lead to CO2 washout and respiratory alkalosis in the neonates. Yes, also feeding difficulty, lethargy, vomiting and constipation, but tachypnea is the most important out there. Also encephalopathy and hyperammonemia obviously will be seen. Yes. Moving on to the treatment. Okay. Moving on to the treatment. treatment. What is the treatment? Yes, the first line of treatment is arginine. First line of treatment is arginine. Why this first line of treatment is arginine? So first of all, it's an essential amino acid. Arginine is an essential amino acid. It is an essential amino acid. Yes. Secondly, it's a provider of ornithine. If ornithine gets blocked, ornithine can be provided from arginine. Also, it's a positive regulator of N-acetyl glutamate. Positive regulator of N-acetyl glutamate, which favors CPS1 enzyme, carboxyl phosphate synthetase rate limiting enzyme is favors, and it is arginine is a positive regulator of the N-acetyl glutamate. Yes, this is why arginine is used, but arginine is also contraindicated. Arginine is contraindicated, or means not used in arginase defect cases. In that case, arginine will not be used and gets accumulated in arginase defect. Yes, in this one condition, arginase defect, arginine is contraindicated. Yes. Also, so the first line of treatment was arginine. The second line of treatment we'll see is our acetylation therapy. Acetylation therapy. Yes, acetylation therapy, we use sodium benzoate and phenyl butyrate. Sodium benzoate and phenyl butyrate. Okay, now why phenyl butyrate is used? Why phenyl butyrate? Yeah, the answer to this question, why phenyl butyrate is used, is, see, it's a prodrug. Phenyl butyrate is a prodrug. That means in the body it is metabolized to phenyl acetate. Phenyl butyrate metabolized to phenyl acetate. Okay, now this phenyl acetate can combine with glutamine, and we know this glutamine is a carrier or transporter of ammonia in majority of the organs of the body, including brain. We discussed in the general amino acid discussion. It's a carrier of ammonia, can combine with glutamine, phenyl acetate, and remove ammonia from body through this glutamine. It's a carrier of ammonia. Phenyl butyrate is converted to phenyl acetate. That's why it is used. And also phenyl butyrate is scavenging ammonia here. Okay, so phenyl butyrate is scavenging ammonia. Yes. Now this sodium benzoate we have discussed, sodium benzoate and phenyl butyrate. Sodium benzoate can combine with glycine. Combine with glycine, okay, to form benzoyl glycine. benzoyl glycine. Sodium benzoate with glycine to form benzoyl glycine. This is also scavenging ammonia. Scavenging ammonia. Yes. Benzoyl glycine, sodium benzoate with glycine. Here phenyl acetate with glutamine removes ammonia. This is the acetylation therapy in which sodium benzoate and phenyl butyrate are used. So this was a total discussion on urea cycle. So we have seen the enzymes, steps in mitochondria and cytoplasm, what all occurs. Then the enzyme defects will lead to which all disease, their features, their names, and finally we see the treatment. Treatment was, first line was arginine, second line was acetylation therapy using sodium benzoate and phenyl butyrate. So very important topic in amino acid metabolism. It's an entry point. So please do revise them, guys. Thanks for watching. I will catch you in the next one. Good luck.